vocês aqui. E sempre muito bom, né, ter esses momentos de trocar um pouco de experiência e conhecer outros trabalhos, né, na área de genética, melhoramento e biotecnologia de batata. Então, hoje, né, também, né, agradecer especialmente ao Guilherme, né, que é o nosso convidado, e que vai compartilhar toda a experiência que ele tem nessa área de genética e estatística, melhoramento molecular em poliploides, dando exemplos, então, em batata e batata doce. Eu vou fazer, então, uma introdução aqui do Guilherme, né, e também, assim, o seminário, ele deve ser em torno de, acho que, Guilherme, 30 ou 45 minutos, né, e aí depois a gente vai abrir para perguntas, e as perguntas podem ser da oral, né, aqui direto, ou então podem já ir escrevendo no chat também. Então, Guilherme, muito obrigada, né, por estar dividindo conosco aqui esse momento de integração entre o pessoal da área que trabalha, né, com batata, e também alguns convidados da batata doce, né, já que está incluído aqui no título, a gente também chamou, né, convidou alguns colegas que trabalham com melhoramento de batata doce. O Guilherme, então, o Guilherme tem bacharelado, né, em licenciatura em Ciências Biológicas em 2009 pela Universidade Estadual da Paraíba, e é mestre em 2011, doutor em 2015, em Genética e Melhoramento de Plantas pela Escola Superior de Agricultura Luiz de Queiroz, a ESALC, né, da USP, Universidade de São Paulo. Realizou pesquisa de pós-doutorado, né, entre 2015 e 2019 na North Carolina State University. Foi bolsista do programa de internacionalização, né, o PRINT CAPES USP. Jovem talento com experiência no exterior da ESALC, 2019-2020. E pesquisador também do Centro Internacional de La Papa, do CITE, 2020-2021. Atualmente é professora adjunto da Universidade Federal de Viçosa, tem experiência em desenvolvimento e aplicação de ferramentas moleculares e genético-estatísticas no melhoramento de plantas cultivadas, coordenando, então, o Grupo de Melhoramento Molecular do Departamento de Agronomia da Universidade Federal de Viçosa e atuando nas áreas de genética molecular, genética estatística, genética quantitativa e bioinformática. O Guilherme também tem vários orientados, né, de graduação, pós-graduação, mestrado, doutorado, pós-doutorado, e tem uma grande experiência nessa linha, então, de genética estatística e aplicação, então, de ferramentas moleculares aplicada mesmo ao melhoramento genético de poliploides. E hoje vai, então, compartilhar conosco a experiência dele com essa palestra, que é Genomics-assisted breeding in inautopolyploid crops, experiencesfrom potato and sweet potato. Guilherme, obrigada. E, pessoal, vocês podem, então, por favor, irem colocando as perguntas aqui no chat, ou então, ao final, também a gente vai ter um momento para discussão. E, mais uma vez, também agradeço vocês que estão aqui conosco, né, fazendo parte desse momento aqui de troca de experiências. Guilherme, tudo contigo.
Obrigado, Carol. Eu penso que eu devo falar inglês, certo?
Certo, acho que tem várias pessoas aqui de espanhol, né?
All right, so I'm going to switch to English, but the questions and the comments can be either English, Portuguese, or Spanish. I just believe it's going to be easier because we understand better Spanish, right? Portuguese speakers understand better Spanish than the opposite. So I believe English is going to make it more accessible. But if you want me to switch back to Portuguese, I can do it, all right? So thank you very much, Dr. Carolina Castro, for the invitation, the Association Latino-Americana de la Papa. It's also amazing to talk to different people that work with us or in parallel with the same species, right? I decided to bring this topic here because it's what we are working with for almost 7 to 8 years now. Focused on sweet potato at first, but soon we realized that this could also be applied to potato breeding. And I'm going to bring examples from both of them, right, of these genomics-assisted breeding implementation. But before we get started, I wanted to talk to you about polyploid crops, and what it makes it different when comparing to diploid crops when we are trying to employ and use and develop molecular markers, right? So we are briefly going to mention the idea of dosage calling, and then finally give you examples of applications in the GAB implementation, right? So linkage mapping, QTL mapping, marker-assisted selection, and genomic selection, right? So as Carol mentioned, this all started with at least for me, right? It started with the genomic tools for sweet potato project. I was a postdoc at North Carolina State when the project started. And the main goal was to develop these tools, right, to facilitate sweet potato breeding in sub-Saharan Africa. And we are proud to say that now we can do it, right, in sub-Saharan Africa in several countries. During that time, we partnered with several institutions, some of them focusing on the population development and phenotypic data collection, right? So International Potato Center and National Crops Resources Research Institute in Uganda. Other universities in the US and Australia were responsible for the genomics, transcriptomics, and bioinformatics. There was a team at Cornell responsible for the sweet potato database, and that's also building it for potato. And we at North Carolina State University were more focused on the genotypic data and statistical genetics analysis, right? So we did the project, we finished it in 2019. Then we were able to get it renewed, now under the name of Sweet Gains, Genetic Advances and Innovative Seed Systems. So this one ended in January this year, and now we are starting RTB breeding project until the end of 2024. So I believe that everyone knows the importance of sweet potato, but I just find these pictures very illustrative of what it means. So some countries have sweet potatoes as a staple food because they are rich in carbohydrates, dietary fiber, vitamins, and minerals. But the preferred variety for a long time in Africa was the white fleshed one, right? Or the cream fleshed ones. And the lack of vitamin A in like pregnant women and child, right, and children is dangerous to their development. And they decided like to include, right, another source of vitamin A precursor, the beta carotene, in such a staple food, right? So this is a breeding program that has been carried out for a long time. It was awarded as a World Food Prize in 2016, if I'm not mistaken. But at that time, people were basically doing traditional breeding, right? Conventional breeding. So the goals of these new projects is to modernize them, right? All right, so talk briefly about the polyploidy in sweet potato. We have a picture of the chromosomes of Ipomia trifida, a diploid relative of sweet potato, which has 30 chromosomes, right? Whereas the hexaploid sweet potato has 90. So it's a hexaploid species, right? We can see how it increases in terms of complexity. Of course, sweet potato is not the only polyploid crop we cultivate, and it's important nowadays. We have several other examples, such as the potato, as you know, sugarcane, fruit crops like kiwi and blueberry, ornamental crops like rose and chrysanthemum, and forage crops like alfalfa and guinea grass, right? This is one type of polyploid. We call them inautopolyploid. In the allopolyploid session, we have several other crops of importance, right? Several seed crops, coffee, cotton, and or also other forage crops. So understanding the genetics of polyploid species is difficult, of course, but we need to do it because of the needs for implementation of these genomic tools, right? Because we are talking about potato and sweet potato, I'm going to focus on the autopolyploid type of analysis, right? For allopolyploid, something changes, and it's kind of in between a diploid and an autopolyploid. But for example, doing dosage calling, we realize that it's also important, right? Which was something that people are not doing for allopolyploid species, but now we notice the importance, all right? So just to make it clear, right, how it's the difference is going to impact our analysis. So in the polyploid classification, we have two types, right, as I was mentioning. The allopolyploids that are originated from 2 or 3 different species, right, in such a way that their chromosomes in an allopolyploid species is going to be preferentially paired within the genome of origin, right? So this is just an example of wheat, right, where 3 species contributed with their chromosomes, right? And when we do cytogenetic analysis, we can actually realize their differences, right? Whereas in autopolyploid species, such as potato, right, we cannot distinguish the set of chromosomes. So all the marks, the cytogenetic marks for chromosome 1, for example, they're going to look like the same. And that has to do with the fact that the chromosomes, duplicated chromosomes, were originated from the same species, right? So we do not see what we see in allopolyploid species, which is preferential pairing, right? What we see is random pairing. And this is going to impact first the segregation, right, in terms of molecular markers, as well as the way we analyze the data jointly, right? Another consequence, as I was saying, of these chromosome behavior is in terms of inheritance, right? So we have allopolyploid species mostly doing what we call disomic inheritance, right? Where 2 homologous chromosome pairs pair, right, forming bivalent. Whereas, and that's what we call preferential pairing, whereas the autopolyploid ones are able to form multivalent, right? So more than 2 chromosomes can pair at the same time. Like most of the times they are gonna pair as bivalents, right? 2 bivalents, but they can also form quadrivalent or 1 univalent and 1 trivalent, right? This is the case of an autotetraploid, all right? So let's start seeing how this is going to impact our analysis and why we should pay attention to the type of the classification of the polyploid crop we work with. In the case of a diploid species, as we know, right, and dealing with the biallelic locus, right, like the one characterized with a big A and a little a, right, we have gametes forming half of the gametes containing big A and the other half of gametes containing little a, right? In autotetraploids, because of the random pairing, we have another distribution of these alleles, right? Now, for example, if we have 2 copies of the big A and 2 copies of the little a, we are going to have a gametes with these proportions here. So 17% of the gametes are going to be carrying big A, 67% of the gametes are going to be carrying big A, little a, and 17% of the gametes are going to be carrying little a, right? And of course, they have to carry 2 copies of the same chromosome, right, or the same locus, because the zygote or the individual has to come up with 4 copies again, right, after the cross. And these, I forget to mention, but these in diploid leads to a segregation of 1 to 2 to 1, right, as we know. But in an autotetraploid, that segregation is going to be of 1 to 8 to 18 to 8 to 1, right, with these type of genotypes. Now, if we look at allotetraploid species, right, we are going to have the same proportion for each one of the alleles, because again, there is preferential pairing happening, right? So the differences here is just basically in terms of how the alleles are positioned in the during meiosis, right? But that is going to lead us to the same proportion of each one of these alleles. And the type of segregation you're going to see is of 1 to 4 to 6 to 4 to 1. All right, so based on, for example, the segregation, we will be able to characterize the type of crop, right, or species. But one interesting thing, and that's what we are going to see next, is that autotetraploid species are going to form up to 5 different genotypes, right, depending on the type of cross we are doing. So in this case, we are crossing 2 duplex individuals, right? Or 2 individuals that have 2 copies of the big A. And that's the kind of pattern we are going to see. Of course, there are many other options, right, of combining 2 heterozygous individuals which is going to lead us to different proportions. This is just one example. And same thing happens with allotetraploids, right? Different situations. All right, now let's talk about dosage calling, right? So for a biallelic marker like SNPs, right, polymorphism, single nucleotide polymorphisms, there are up to 5 possible classes if we are talking about a tetraploid species, right? So one class is going to be represented by 0, so which means 0 copies of allele big A, or 1 individuals with 1 copy of big A, 2 copies, 3 copies of big A, and 4 copies of big A, right? So we are basically counting the number of copies of one of the alleles, because we are going to know the other allele just by difference, right? The problem is, even though that's what happens in nature, our molecular tools might be limited in assessing this type of information, right? For example, we used to use certain qualitative techniques, like observing bands in a gel, right? Which would make it impossible to distinguish genotypes based on their dosage, right? We would basically see presence for all the individuals that have at least 1 big A, and absence for the individual that doesn't have a big A, right? And that's why the dosage is so important, right? Because it's going to allow us to assess more information about each one of the individuals, right? That we are genotyping with the molecular marker. So usually we use a number of a reference allele respective to the alternate allele, right? And for each one of these classes, we give in literature different names, right? So for the 0 would be the nulliplex. For 1 would be simplex. For 2, which was the example I was giving in the previous slide, we named them duplex. 3 triplex, and 4 tetraplex, right? It's just a naming convention that we use out there. Now, how can we assess or how can we obtain data which is enough for us to evaluate dosage, right? There are several ways. Some of them are based on hybridization, for example, right? In such a way we can measure the intensity of hybridization for each one of the alleles and use this amount of signal, right, to distinguish among the 5 possible dosages, right, in the case of a tetraploid. That's the case of the famous chip array we use in potato, right? This is basically giving us different hybridization levels depending on the allele that that individual carries for each one of the locus. Other technique that allows us to assess dosage is by using second generation sequencing, right? Because we can read the same location in the genome several times in such a way that amount of reads is going to indicate which allele is more frequent for that specific location for one individual, right? So for example, let's suppose this is a tetraploid individual, right? In this last SNP here, we have more or less the same amount of allele green and allele yellow, right? So in this case, I would say that this individual is a duplex, right? Because it has two copies of the allele identified as green and two copies of the allele identified as yellow, right? Whereas this other individual carries less copies of the blue allele in comparison to the green one, right? So in this case, if it's a tetraploid, I would say that it has one blue allele and three green alleles, right? Based on the ratio, right? Based on the amount, the proportion of these two alleles that I have assessed through sequencing, right? Some of these techniques are generally called genotype by sequencing, right? But we have variants of these techniques out there. All right, so what we did when we started working with sweetpotato was to come up with an adapted protocol, GBS protocol for polyploids, which means we would have increased amount of rates that could enable us to call the dosages, right? So that involves several modifications you can see in the papers. But that would basically deliver us with the raw sequencing data. So we need to count the reads, right? Or count the alleles generated by these reads and call the dosage, right? So this is something we can do through a combination of packages, or there are actually nowadays several packages out there that allows us to do this dosage calling, right? From VCF files, right? Variant call formats, which is an output of the variant call, all right? Now, let me show you examples of, like real example of populations genotyped with some sort of GBS, right? It could be variants of that protocol, just so you can have an idea of what we are actually dealing with. So for a species, a diploid species like Hypomia trifida, the diploid wild relative, diploid wild relative of sweet potato, right? We would expect three type of classes, right? The individual that's homozygous for one allele, let's say A, homozygous for the other allele, alternate allele, let's say C, or heterozygous, right? And if we evaluate a biparental population, for example, that crosses one individual fixed for one allele and the other individual heterozygote for their allele, we are going to have a segregation of type 1 to 1, right? So this plot's basically showing us the amount of rates containing one specific allele for each individual. So each dot right here is an individual, right? So this one, for example, has over 400 reads sequences, right, for the same location with the same allele. So I say that it's a homozygous individual, right? It's AA. Whereas this individual here has half of the reads with the allele A and the other half of the alleles with the allele C. Half of the reads with the allele C. So it's a heterozygous individual, right? CA. Now, if I cross two heterozygote individuals, right? AC crossed with AC, we are going to have a segregation proportion like 1 to 2 to 1, right? So in addition to the two, the homozygous for allele A, we have the heterozygous and also the homozygous for allele C, right? So diploid species, three possible classes, right? Two homozygous and one heterozygous class. easy to do for diploid species in general. Now, let's complicate things a little bit, right? And work with tetraploid species like potato. In this case, I'm going to have individuals carrying 0, 1, 2, 3, or 4 copies of allele C, right? Evaluating a diversity panel with 93 or 83 individuals, for example, we are able to identify these 5 classes, right? So it doesn't need to be derived from a biparental cross. It can be breeding panels, right? Breeding populations, diversity panels, in a such a way that if we run those pipelines, right, bioinformatic pipelines, we are going to be able to distinguish individuals based on their dosage, right? Now, what people did for a long time was using deployed tools, right? Deployed bioinformatic pipelines, or pipelines designed for deploys. And what such pipelines would do, they would combine all these individuals here, saying that it belongs to a single heterozygous class, which is clearly wrong from the informativeness standpoint, right? Because we are basically saying that these individuals here that has 1 copy of C, for example, is from belongs to the same class as this individual that has 3 copies of C. But this is what people did for a long time before we had these tools, right? Now, we should target such level of information, right? We need to increase the level of information. Now, another example with the hexaploid sweet potato, right? It's a hexaploid species, so we expect up to 7 classes, right? 2 homozygous, 0 and 6, and 5 classes of heterozygous, through 1 through 5, right? So in an example of a biparental cross, we can see something like this, right? Individuals fixed for 1 allele, but with several heterozygous classes, right? And as you can see, the need of increasing this read depth is imperative, because that's the way we are going to be able to distinguish those classes, right? Because if the number of reads is too small, we won't be able to distinguish those individuals, right? So we need to increase this read depth. All right, so let's move on to the applications, right? We used mainly 2 biparental populations, one from potato and another for sweet potato, to develop the tools we use nowadays for the applications we are going to talk about. So the first population is from potato, a cross between Atlantic and B18-2095, with 154 individuals genotyped with a Illumina chip array, right? More or less for 8,000 SNPs. And then this population was evaluated for 4 years for yield and qualitative traits, all right? The sweet potato population was a cross between Beauregard, the orange-fleshed sweet potato, and Tanzania, a cream-fleshed sweet potato from Uganda. We genotyped and phenotyped 315 individuals. So for GBS poly delivered over 1 million SNPs, right? And the yield and qualitative traits were obtained from 6 environments, combinations of 2 years and 3 locations. So first thing we did was to build a linkage map, right? So the population, the potato population, right, derived a map with over 4K SNPs, right, spanning 1.6 K centimorgans. By using the dosage information and the models we have developed, we were able to recover, right, the information about all the parental homologous haplotypes, right, which is a number of 4 for each parent, right? So A, B, C, D, we are calling the 4 haplotypes of Atlantic, and E, F, G, H, the 4 haplotypes for B18, 29, 5, right? And this is something we can basically distribute our phase or build haplotypes, right, based on the dosage information. And of course, we have these dosages across all these chromosomes, right? This is an example of chromosome 1. Another thing we can do based on the linkage map is to compute the inheritance or see the inheritance of chromosomal segments from parents to progeny, right? So basically, we are going to be able to detect the places where recombinations happened, right, during gamete formation, and that ended up in individuals that we have evaluated. So when we see fragments or combinations of chromosomes, right, so we have 2 of them coming from the female parent, right, and 2 of them coming from the male parent. And when we see a mix of more than 2 colors, that means that at least 3 chromosomes were paired during meiosis. And we saw that several times, right? We see it here, for example, between these 2 homologs, there was a recombination here, here, and here, right? But in at least 3 chromosomes were participating. In this case, 4 chromosomes were participating at the same time for the formation of the gamete, kind of showing the multivalent pairing we talked about previously, right? Some of the chromosomes are not going to go through any type of recombination, right? So their individuals are going to inherit exactly the same chromosome as the parents, right? This is counting the amount of times we realized that more than 2 homologs were participating in the pairing, right, during meiosis. So the light green represents that at least 3 homologs were paired during meiosis, and the dark green represents the amount of individuals, right, generated, or the gametes that were generated from a pairing of 4 homologs at the same time, all right? Of course, bivalent pairing is much more frequently observed, right? But we can also see that trivalent and tetravalent, quadrivalent pairing happened. Another thing we could see was that there was no preferential pairing, right? So all the combinations of pairs of chromosomes were observed at the same level for all chromosomes. If we had seen a preferential pairing, we would see one of these lines far from the expectation, right? Which wasn't the case, for potato at least. And this is also used as a basis for computation, the proportion of alleles identical by descent shared among individuals, which is key for the QTL mapping work, all right, which is what we are going to talk about next. So in this example, after we use, right, the linkage map to see which individuals share the same portion of the genome, we come up with things like this, right? So we are looking at one specific location, precisely the first marker of chromosome 1, and realize that some individuals here, so we have 154 individuals, right, for the potato population, and 36 possible genotypes, right? So some of these individuals are sharing the same alleles, right, which make them identical by descent for all 4 alleles, right? And of course, they're going to vary, right? The proportion of identity by descent, depending on which fragment of chromosomes each individual has inherited from the parents, right? And this is just an example of over chromosome 5. locations where the recombination happened, right? So this specific individual, one single individual had inherited AC from parent 1, FH from parent 2, and then a recombination happened. The change here was from A to D, right? So the individual is for great part of the chromosome C, D, F, H, and then at the end, another recombination happened, now in the other parent, and H became G through a recombination, right? So it's nice because based on the linkage map, we can actually see all this happening. All right, now let's talk about quantitative trait mapping, right? Or QTL mapping. And let's switch to our experience in the field, right? So when we plant full seed populations like the ones we had, right, for potato and sweet potato, we observe a certain variation, right? So this variation here we see is the area under the senescence curve for the foliage maturity, all right? So we evaluated these individuals in the field, and we see that there is a variation. So phenotypic data, and we like to see histograms like this, right? They can be described in terms of mean and variance. So just as an example, the mean here is 74, and the variance is 400, right? Now, what makes potatoes different from each other, right? Because if there is variation, it's because the potatoes are different, right? So let's think about two cases. The first one would be a experiment that has only one single clone. So Atlantic, for example, we planted it and we still could see some variation. Because they're clones, we know that all the variation is because of the environmental component, right? It's basically a residual, something we couldn't control. So there is no genetic contribution, right? Now, in the case of an F1 population, right, a full seed population, in addition to the environmental component, right, we are also going to have the genetic component, which we generally call polygenic effect, right? So the combination of all the genes contributing to the variation of one specific trait, right? Now, our goal with the QTL mapping is to identify specific genes out of this polygene scenario, right? Contributing more, this is what we call major QTL, right? Contributing more to the variation of a trait. So we can say that phenotypic variance can be attributed to different sources, right? And these values, right, of genetic variance and total phenotypic variance is something that we can use to compute, for example, broad sense heritability, right? We look at the proportion of the phenotypic variance that can be explained by the genetic component, right? Now, if I want to detail this genetic component even more, we can use a relationship matrix among individuals, right, to study the additive portion or the heritable portion that contributes to the phenotype, right? We can do it based on pedigree, for example, but we can also do it based on molecular markers, right? In this case, we would come up with a genomic relationship matrix, right? Represented by A here. So once we can separate what has been inherited, right, through gametes, we can calculate things like narrow sense heritability, right? Because now I'm focusing on the additive portion or the heritable effects of the genes, right? So genetic variance can be also decomposed into additive and other variances, right? Like dominance, epistasis, and so on. Now, we build a genomic relationship matrix for 154 full sieves, right? Originated from that cross. And of course, we realized that all of them are around 0.5, right? Which is, in fact, the expected value of relationship among full sieves, right? So this is the realization that we see. Of course, we see different colors. Maybe we have clones out there, right? Some dots in pairs of individuals are marked with red, right? Representing 100% of identity by descent for all oocyte, right? We have evaluated, but most of the times they are only going to be around 0.5, right? Which is the expected for full SIPs. And this is what we call the realized relationship matrix, right? Based on the markers. Now, this was taking into account all the markers or all positions in the genome virtually, right? At least. Now, our interested is in knowing whether there are any loci associated or locus or QTL, right? Associated with the phenotype. And that's when we build specific matrices for every position in the genome. And we do so because now we are interested in that specific variance component that's associated with that location, and that explains a lot of the phenotypic variation, right? And why do we do that? Because we are interested, we know that in the region of a gene influencing a quantitative trait, right? Correlated QTL effects among individuals are due to shared alleles identical by descent, right? There is a gene, and some individuals share the same allele, they are going to be more similar, right? Phenotypically speaking, for the trait influenced by that specific region. So let's see how we compute the identity by descent, right? And again, we are going to start with the diploid crops. We have two gametes in each parent, and each parent is going to produce two different gametes, right? When we cross these two different gametes from each parent, we are going to have a set of 4 genotypes, possible 4 genotypes, right? Each one of them with the same frequency in the population. Now, if I want to count for every pair of individuals, if they are completely related or not for that specific position, I just need to look at their alleles, right? The alleles they inherited from the parents. So if they inherited A and C and A and C, so they share 2 out of 2 alleles, right? 100% of individual of identity by descent. Individuals can share only 1 allele, right? Identical by descent, or 0 allele identical by descent, by chance, right? So this is the case of a diploid. In the case of a hexaploid, we expect the tetraploid, sorry. We expect that each individual is going to generate 6 different gametes, right? Considering that all 4 haplotypes are different. And in this case, if we put together 6 gametes from each parent, right, we are going to end up with 36 genotypes. And again, we can study whether a pair of individuals share 0, 1, 2, 3, or 4 alleles identical by descent, right? Again, how? By looking at the genetic map. In the case of a hexaploid species like sweet potato, we are going to have each individual generating 20 different gametes, right? And up to 400 genotypes after the cross. And again, we can, based on the information derived from the genetic map, say whether a pair of individuals share 0, 1, 2, 3, 4, or 5, 5 or 6 alleles identical by descent. All right, when we did that for the potato population and for that specific trait we were talking about, we identify one major QTL at least on chromosome 5. Maybe some of you know it already. When we look at the genomic relationship matrix for that specific QTL, this way with a unordered trait values, we do not see any patterns, right? But when we move individuals that have the same phenotypic value close together, we can see that they share more alleles identical by descent, right? So it's more red here around individuals that are closely related phenotypically speaking, right? Results from this population in general, right? So we were talking about these major QTL for foliage maturity in this population that turns out to be co-located with several other QTL on chromosome 5 as well, right? Like yield and internal heat necrosis. We also did similar, and I'm going to go through the results for sweet potato very briefly. We also did the same analysis for the Borregat by Tanzania population, right? Build the genetic map. Now 6 haplotypes from each parent, right? And we use that again to compute identity by descent among individuals, right? Identifying where the recombinations happened and ended up in each individual for every location, right? So we used this map to map QTL for several traits like dry matter, beta carotene, flash color. And also, interestingly, we identified co-located QTL for these traits on chromosome 3, right? Especially in this location, and also on chromosome 12 for this location. Another very strong major QTL was found for skin color, also on chromosome 12, but in a different location, right? So what we decided to do was look at these regions, trying to find the genes involved in these processes, right? And design SNP markers, Cas markers for performing marker-assisted selection. For example, for the flash color or beta carotene content, right? We identified 2 genes around that region. So phytoin synthase, which is a gene involved in carotenoid synthesis, and also sucrose synthase, which is involved in starch biosynthesis, right? These 2 traits in sweet potato are negatively correlated. So when we study when we study the contributions of each one of the alleles, we see that it increases beta carotene content. At the same time, it decreases dry matter. And our interest is actually to have them both increased in sweet potato, right? Now we realized the basis, the genetic basis of this correlation, right? Which is basically based on the linkage group 3. So in, for example, right, for dry matter, the same allele, the same haplotype that decreases, right, the value for dry matter or starch, it's the one that increases the one for beta carotene. But this is not something we want. But in fact, because the 2 genes are linked, very closely linked, right, they are segregating together and causing that negative correlation. Same type of study we can do with the QTL on chromosome 12. Now, doing GBS, right, for, having many of SNPs, we realized very soon that it was not going to be needed to build such genomic relationship matrix or build linkage maps, right? So people at Reading Insight created a dark tag panel that has up to 3.5K SNPs. And in populations in Africa, for example, they show polymorphism for 2.7K, right? Very well distributed for sweet potato. We can see population structures and things like that. And we can even use it for GWAS, right? Genome-wide association analysis. In this case here, we can see the same peak on chromosome 3 and the same peak on chromosome 12 for flesh color and skin color, respectively, based on this small panel of 2.7K SNPs, right? Which is nothing compared to 1 million that we have used, but still can deliver the same type of answer, right? Now for a breeding diverse population, not only a biparental population, right? Another usage we did for speaking about potato for this type of panel, right? The dark tag panel was realizing 2.3 polymorphisms for this breeding population and clearly seeing the structure in population caused by the usage of four males, right? So one male, another male, they have big groups here, and the two other males here, right? So it's easy to assess this type of information nowadays based on the information from 2.3K SNPs in potato. We also looked at the distribution of the SNPs and tried to find genes, a major QTL for traits like flower color, and we found it on chromosome 2 for potato. All right, now moving to very quickly to the marker-assisted selection, right? We decided to look at the genes associated with those major QTL on sweet potato, designed Cas markers, and used this technology to assess dosage in a breeding population, right? When we assess dosage in this type of population, we see something like this, right? So up to 7 different colors when we are talking about sweet potato. Now, I'm just going to show the tests of a couple of SNPs that we are currently validating in this population. So for a skin color, we can see an I-square of 0.49, right, and an additive orientation of this SNP, right? So this is exactly where the QTL was located on chromosome 12 in the case of skin color, right? So very nicely located, which means that, for example, skin color is an important trait for sweet potato for sure, because that's what people see in the market, right, when they're picking their sweet potato. And now you can evaluate the color of the sweet potato, right, the skin color, before it even gets roots, right? We can evaluate based on the DNA. Maybe this is not as an interesting application, but we also did the same work for nematode resistance. And if you have worked with nematode before, you know how hard it is to get this type of phenotypic data, right? So we also did the conversion. So we found the QTL, and we also did the conversion of this SNP associated with nematode resistance, and converted it into Cas and validated into a population, now showing more of a dominant behavior in terms of resistance, right? This QTL in sweet potato was mapped on chromosome 7 for myeloidogeny incognita. All right, now just to finalize with the genomic enabled prediction, I'm going to show you some of the tests we made, still using biparental populations, just to check the importance of using haplotypes or dosage for genomic selection, right? We are using basic blood-derived models, right? So based on the mixed model approaches, our G matrix is exactly the genomic relationship matrix, based either on the full set of marker, right? That's what the approach we are calling GBLUP, or based on the QTL identified for every training set population. So what we found out when we compared QBLUP, right? So prediction based on QTL that we have identified for each one of the training sets, and GBLUP, was that for quality-related traits, so more type of oligogenic traits, right, with traits controlled by major QTL, we realized that doing prediction based on QTL is better, right? We can predict it better in comparison to the approaches that use the whole data set, the whole market set. Now, when it comes to yield-related traits, we cannot see such a difference, right? So they are more or less at the same level, which has an explanation, right? Yield-related traits are more polygenic in nature, right? So they have many more genes, and each one of them is contributing just for a very small proportion. We cannot detect as a QTL without making a lot of mistakes, right? So applying GBLUP that's just as good as running a QTL model in general. Now, we were also interested in comparing the market informativeness. In this case, we are focusing on GBLUP models. And then we compared the building genomic relationship matrix based on diploid-like markers, right? So 0, 1s, and 2s, dosage markers 0 through 6, and haplotypes, right? The haplotypes we have built using the linkage mapping approach. So one thing we notice is that for all traits, it's much better to use at least dosage markers, right? The dosage information for the markers. Because if we place all the heterozygotes into one single heterozygote class, we are going to miss a lot of information, right, in terms of prediction. Now, the haplotypes only increased the prediction for some of the traits, right? So this is the example for the quality-related traits, and we have also the same type of observation for yield-related traits, right? Using dosage always increase the predictive ability in comparison to diploid-like markers, right? And having haplotypes involved would increase some of the traits, but not all of them. Now, just some final remarks. We have to find the right tools, right? As you noticed, using diploid made software and tools and markers, right, is going to make it it's going to lead us to several issues, right? Like the ones we related with the predictions. In general, haplotypes are going to be more informative in comparison to dosage, but dosage is way better than having just diploid-like markers, right? Another thing we noticed, like when implementing these type of tools, right, is that the turnaround time matters. So we had to come up with some technologies, right, that would be available for the breeders very quickly, so they can make their decisions in the fastest way, right? Another final message is that we need to get to know our traits and strategize, right? So what we are doing for sweet potato at SIP is focusing on marker-assisted selection for oligogenic traits and selecting them in early stages, right? Like for beta-carotene content and nematode resistance. And use genomic-enabled prediction or genomic selection for more polygenic traits, right? And selecting them in later stages, like for root yield and texture after cooking, all right? So I would like to thank you for the attention and all the groups involved in, data collection and analytical support, my group at UFV, as well as the financial support of Bill and Melinda Gates Foundation, right, that supported all this work, and scholarships we get from Brazilian foundations. All right, thank you very much.
Obrigada, Guilherme. Pessoal, Guilherme, muito bem didático, né, e acho que traz aí coisas novas, né, e para a gente aqui começar a pensar, né, na aplicação disso nos programas de melhoramento. Eu abro aqui para perguntas. Alguém gostaria já de começar a fazer teria alguma pergunta ou comentário? Bom, enquanto o pessoal vai pensando, eu vou fazer uma pergunta. Vou fazer em português. Acho que a maioria daqui está... Se tiver algum problema... Gustavo, tem algum problema de ser em português ou prefere em inglês? Guilherme, para quem está começando, tem um programa de melhoramento de batata já instalado e gostaria de começar a ter alguma ação como ou seleção genômica, que porque eu acho que para essas características, né, como tu comentaste, características mais quantitativas, eu acho que é um gargalo bem grande, né, que tem nos programas de poliploid. O que tu recomendas, assim, desde a escolha da população, do tipo de marcador, você poderias comentar um pouco com relação a isso?
Uhum. Eu respondo em português mesmo, talvez?
Elisa, what do you think, Elisa, in Portuguese, the answer, or do you understand the question, please?
En portugués, sí, está bien. Perfecto.
Ok. É uma boa pergunta, Carol, obrigado. É interessante, né, porque todo mundo tem interesse em fazer esse tipo de aplicação, mas sempre esbarra, né, nessa dificuldade em obter os dados moleculares primeiro, né? Então, boa parte desses trabalhos, na verdade, estão providenciando esse tipo de ferramenta, né? Hoje em dia se torna mais barato, o tempo de entrega é relativamente rápido, né? E os programas não precisam nem ter laboratório para extrair DNA, porque hoje em dia a gente manda as folhas, né, dissecadas para as empresas prepararem, né, esse conjunto de marcadores para a gente. E algumas delas vão entregar, inclusive, a parte de bioinformática, já com as dosagens, né, alélicas para cada um dos SNPs. Eu diria que se a pessoa ainda tem dúvida, né, se o melhorista ainda tem dúvida se essa vai ser uma boa oportunidade para ele ou não, eu diria para começar a genotipar as populações o mais cedo possível, nem que seja para ter melhores predições, né, para ter melhores BLUPs na hora de fazer seleção. Não precisa ser nem seleção genômica, é só uma predição mais informada, né, em termos do compartilhamento, né, de alelos entre indivíduos, que reforça essas indicações, né, de melhores genótipos ou melhores genitores de modo geral, né? Então, agora, se a pessoa quiser, né, porque isso tem isso também, né? Seleção genômica envolve mudanças no pipeline de melhoramento também, né? Não dá para continuar fazendo exatamente o mesmo melhoramento que se fazia antes, uma vez que se decida, né, fazer seleção genômica. O meu conselho é, começa a passar marcador, nem que seja para reunir dados, né, em populações, né, que a gente costuma chamar de intermediate yield trials, por exemplo, para ter dado, né, de produção e dados de caracteres qualitativos suficientes para treinar um modelo mais para frente. Mas se a pessoa decidir desde já fazer parte de seleção genômica, né, ela vai precisar alterar um pouco as populações, né? Por quê? Modelo de seleção genômica sofre um forte viés em função da representação dos genitores. Então, por exemplo, se é um programa de melhoramento acostumado a utilizar policruzamento, polycross, né? Você pode estar gerando um viés enorme para alguns determinados genitores masculinos, né? Para o feminino, a gente consegue representar bem, né? Porque é de onde a gente está coletando as sementes. Mas para o masculino, a gente, como não tem controle do pólen, né? Pode ter uma tendência de enviesar muito o treinamento do modelo para um ou dois genitores masculinos, e a gente está interessado em representar, às vezes, 10, 15, né? Então, pensar no tipo de população, né, na garantia de representação dos genitores dessa população de treinamento seria talvez uma das partes mais fundamentais, certamente, né? Mas um monte de coisa que a gente deveria pensar, garantir, né, essas delineamentos experimentais que entregam um grande poder, né, na hora de obter essas médias fenotípicas para treinar um modelo, né, é super importante. Eu diria assim, que tendo a parte molecular relativamente resolvida, né, do ponto de vista de você pode confiar numa plataforma que te entrega informação suficiente, né, informação que você precisa, que é a dosagem, e de uma maneira relativamente rápida, né, o próximo passo é justamente começar a fazer. Então, começou a fazer e notou benefício em melhorar as predições com base na informação derivada de marcadores moleculares, daí parte para a questão de planejar corretamente essas populações de melhoramento, né, de treinamento, pelo menos, precisam ser bem planejadas, né? Garantir a associação sempre entre população de treinamento e seleção, né? Se a gente está usando genitores muito diferentes, a predição não vai servir. Então, precisa tomar cuidado com algumas estratégias, né, do ponto de vista de programa de melhoramento, mas se a pessoa tem dúvida, começa a reunir esse tipo de informação que eu garanto que não vai se arrepender. Agora, conseguir fazer isso de uma maneira rotineira, né, como a gente tem feito com os parceiros da África, né, aí vai requerer um certo tipo de financiamento em longo prazo, né.
Obrigada, Guilherme. Julieta, do you understand the answer in Portuguese, or do you like to, maybe Guilherme can summarize in English?
No, I don't understand Portuguese, but
Sorry.
there are some words that I can like hear, translate, but not much.
So yeah, very briefly, just to let you know what we are talking about. I was talking about breeders doing it with the populations they are having the field, right? Just to make sure they have better predictions, better blocks, right? Based on molecular markers. This is going to increase the accuracy of selecting parents, for example, it's going to make it more accurate. And the other thing is, if you decide to go with the genomic prediction approach, you'd have to rethink the populations, right? And I was telling the example of polycrosses that tends to bias towards certain male parents, right? We can guarantee the representation of females, but not always of the males. And if the training population is biased, our selection is always going to be biased, right? Making sure that we keep the relationship between training and selection population. In general, we do not have many problems with that. But yeah, trying it at first, making sure you have resources to make it routinely, right? It got cheaper. It's really fast. You do not rely on bioinformaticians anymore to give you the dosage information you need. So yeah, you have everything in hand, at least what these two crops we were talking about to start doing it now.
Sorry, Julieta. Any other question or comment? Elisa?
Sí. Guillerme, what do you think is the challenger in the Latin American region to implement in the breeding program this kind of the tool?
So I don't see the problems in Latin America to be bigger than the ones we have in Africa. We still rely on third-party companies, right, to do the DNA extraction and the molecular markers for us, right? And it's doable. So we are doing it there. Maybe we can think about training, right, or capacity building, or something like that that would make people more, to feel more safe to use such type of tools, right? So training, I guess, it's a big part of it for one person to decide to use, molecular markers into their breeding programs. So I would say maybe the major limitation is like people understanding the benefits of doing it, right, and how it can accelerate the process and making it, more accurate and deliver the genetic gains we hope in a shorter term, right?
Thank you, Guilherme. I have one comment. Elisa, would you like to talk something? To say something?
No.
No, okay. Just one thing I was just thinking now. Do you have any your knowledge about people that are using genomic selection, like in the US, for like in the routine of the program? Do you have this experience and its work, or how hard is it? Because looking all this, it's beautiful, and we see that we are most close to
Right.
use it than in the past, but it's still a little bit, for my impression, very complicated.
Right. Yeah, lots of the potato breeding in the US are made inside the universities, right? Of course, there are companies, but we hear more about the breeding inside universities. And I would say I was part of a meeting like a couple of years ago where there was someone saying, no, you should start using what does it need? Let's try to come up with a way that everyone can do it. And nowadays people are doing it, right, routinely in their breeding programs in the US. One major limitation was the genotyping platform. So people were relying on the chip array, but the chip array was really expensive, right? Getting the information per sample. But then that's why they decided, right? SIP and the University of Wisconsin decided to come up with the DARTech panel for potato, and it's now cheaper to get the data point. And when things get cheaper and the bioinformatics gets done by the company, right? You do not need to do it yourself. It got much more acceptable, by the teams. So yeah, I'd say training is a big part again, right? But then they have their meetings, right, with the objective of creating this capacity building for analyzing such type of data. But it involves a learning curve, right? But it's doable with the knowledge we have in terms of diploid species, right? We just need to add different tools here and there to be able to use this type of data. But in general, it's very doable. I would say that training has a lot to do with it.
Okay, thank you, Guilherme.
Thank you.
Any other question or comment? Guilherme, would you like to comment something?
Yeah, would just like to thank you all. You have my email. You can contact me if you need any assistance. Right, we are working with Carol at Embrapa. We are working with some other folks in other countries, and it's working well, I'm guessing. Right, of course, we are going step by step, but I believe it's doable. And just one final message, right, for the students who are out there and for the advisors, right? If you need training on this type of thing, my lab is entirely dedicated to genetic data analysis, and we are open to receive you as masters and PhD students, right? Especially in Latin America, some of the countries do not have PhD courses in genetics and plant breeding, right? And in Brazil, you can get scholarships from the country, from Brazil, right? To spend 2 years for masters and 4 years for your PhD course, right? So if you are interested, we have it open now. I think it's targeting Brazilians the first time, and then we are going to open for internationals also, right? So if you know anyone interested in pursuing their master's or PhD, please feel free to share this information. All right.
Great.
Thank you, Guilherme, and thank you, everybody. I think it always is a moment to know more and to share experience, and also to see how far we are or we are not to implement this, but we need to start. So I think that these meetings is a very, thank you, Guilherme, because it's a moment that we can all, standardize the information and know what's going on. And thank everybody that's here and making part of this meeting. Elisa?
Yeah, thank you very much, Guilherme. I think we need to thinking about the new opportunities, the implementing this kind of technology in our breeding program. But my feeling is that we need, probably not all the breeding programs are strong to implement it quickly. So I think probably we need to try to create a network that that bring the information. Probably they don't have informaticians, and also we can use like a service, but in the level to the region, because not all the breeding programs have the resource to support this kind of the implementation, this kind of tool and the infrastructure. So I think probably with Caroline, also when you come here and see, we can discussion better and try improve this kind of network for the breeding program they want to interest into the application of these tools. I recognize to Gustavo is in Uruguay. I know he had a material from SIP, the potato breeding material. So, but why not he can implement this kind of tools in our population? So this is only ideas, but probably we need to have other time to have this kind of discussion. Is also Francisco Villaró in Uruguay. So they are, how do you say, a lot of strong people to know very well the breeding programs in the part to the traditional methodology. Right. So we need only to somebody to you, Guille, who create the bridge between us. No, this is my point of view. Thank you very much for your time again. Sorry for my English; it's not very well understood. But thank you again. And yeah, if somebody have this is Ikra.
Yes, I'll send you by email.
Yeah. Ikra have a question. This can you share your give? Yeah. Okay. Oh, great.
I put it right there, and I also shared the link with because we were talking about the U.S., right? So they organize every year a training for people working with polyploid species to learn how to use this type of data and the tools we talked about. And it's very consistent. It happens every year in January. So they get together as a group, get updates, and these are the breeders, right? Learning how to use the tools and applying them to their breeding programs. And maybe this is something we need to target in Latin America, right? Make sure we have this type of training, this type of capacity building, because I think this is going to encourage people more to use them.
Thank you.
Thank you. I can see people from Embrapa Vegetables, Embrapa Hortaliças. They are a team.
Nice. Thank you, guys.
Thank you. Bye-bye. Thank you.
Bye-bye. Thank you. Gracias, Caroline.